Hi guys, welcome to Biology Videos at TDA. This is suitable for Year 12 and Year 13. It's an end of Year 12 paper that I'm going to go through, and a bit of revision and quizzes and things as well. So let's get started. Get a pen and a paper as usual, please. And there's be a lot of pausing, so get ready to pause and have an attempt, because I won't be really teaching very much, I'll be more just revising things with you. Okay, so let's just start off with some interleaving questions. So, question 1, explain countercurrent flow in fish gills. Okay, so the key concept of getting the answer here is the direction in which the water is flowing over the gill lamellae is opposite to the direction of the blood flow. So in this image, the blood is flowing from the 1 to the 9, and these numbers are trying to represent the concentration, an arbitrary unit for concentration of oxygen. Now what this allows us, so the usefulness of this, is it allows us to always have, if you look at the number in the water, it's always 1 above the concentration in the gill lamellae. So therefore, all the way along the gill lamellae, you've got a concentration gradient that's maintained all the way along, and I can show you in a different image. Again, same idea, you've got the blood flow going one way and the water the other, so this is deoxygenated blood going to oxygenated blood. And that then means that all the way along you have this concentration gradient from which oxygen can move from the water into the capillaries. And then if you represent that on a graph, you'll see that although the blood oxygen saturation is lower than the water, but it's maintained all the way along, which is more efficient than the same direction. Okay, I found this image. I thought we could try and do some labelling. So pause the video, maybe just list down on your page in order what you think they are. Okay, and here's the answers. I'll let you check it, pause it and check yourself. Okay, let's do some chapter 8 questions. Explain hydrostatic and oncotic pressure. Okay, so in simplicity, hydrostatic pressure is the force of fluids pushing out of arteries, and it doesn't have to be arteries. This is just the example I've used, but it could be within xylem vessels. And oncotic or osmotic pressure is the pressure of the pull inwards. So you have the pressure of the fluid outside of the capillaries pushing inwards. So some true or false now. Xerophytic plants have no stomata. False, they do have stomata, they just have a lot less because they live in water-starved conditions. Do tree trunks expand at night? True, they do, because the transpirational pull is much less, so the water kind of sits in the trunk, and you can go ahead and measure some trees if you like, but the idea is the transpirational pull is slowing down because the stomata is closed and photosynthesis is slowing down. The Casparian strip prevents water travelling along the cell wall into xylem. True, it forces it out of it. Next one, the apoplastic pathway takes place within the vacuole of cells. False, that is the symplastic pathway. Apoplastic is in the cell walls. Plants respire during the day. True, there's a common misconception that plants do photosynthesis during the day and respiration during the night. They do both in just varying different levels. Photosynthesis will massively slow down at night time when light levels are low, but respiration will be going on throughout. Stomata are only located on the underside of the leaf. False. There are many more in most plants, particularly herbaceous ones. They'll be highly concentrated compared to the top, but there are also some on the top of the leaf, there's also some on the stems themselves. O2, CO2 and water travel through the stomata. True, that's where gas exchange occurs, and we also have the transpirational water loss. Insects have a large surface area to volume ratio, and that's to reduce water loss. False, that large surface area to volume ratio doesn't reduce the water loss, it's going to increase it. It's going to increase everything lost. So large surface area to volume ratio is always going to increase the rate of exchange of something. Wind and temperature increase the rate of transpiration. True, they both do, okay, along with reducing humidity. The symplastic pathway takes place across the cytoplasm of cells. True, in the way that I remember this is symplastic, sounds a bit like symplastic is cytoplasm, that's the way I kind of remember it. Now don't mix this up with cell wall. but SYMCY. Maybe it's the Y that's keeping me kind of remembered there, but that's the way I remember it. Symplastic is in the cytoplasm. Okay, so now I've done a bit of a starter, we're going to do an actual review and exam paper. I won't do the whole thing, so you've got an option now. You can either pause the video now, go to the website, have a go at the full paper maybe over the next 2 days, or 2 hours of lessons, mark it, and then come back to me. If not, I'm going to pick out some common questions, ones we've done recently, and then talk through them, particularly ones that I think are more difficult. A couple of years ago I gave this paper to some year 12s and 13s, so I can kind of remember which ones were weaker for most people, and I've read the exams before. Okay, right, let's have a go. So, I'm only picking out some of the questions. So, question 2 I just thought would be a good one. Autoimmune diseases are often treated with a course of antibody injections. So there's a couple of really important information here if you're having a go at this. First of all, this is saying injections. This is not saying vaccination. So we are injecting an antibody directly. Okay? So which one is true for the immunity arising? So good, it is passive. So the idea is passive. So it's being injected, so it's not natural, it's not from, passive because you are not making the antibodies, that's why it's passive, and it's artificial because you're being injected. So the idea is passive because you're having the antibodies injected into you. If it was active, your body would be responding and making the antibodies themselves. Next one, I quite like Daphnia. This image wasn't on the exam paper, but I put it on there to kind of get to know. Now this is going to be really common, and I like this question, because it's giving you information about something you should have learned and you're applying it to something new. So the thing you should have learned is hemolymph, and you've learned that in regards to insects, okay, locusts and things like that. Whereas in this example we've got a Daphnia magnum. Now that's a water flea, it's a particularly large one. It's good to look at under the microscope. You can see all the various features. This is kind of what it will look like under a pretty clear microscope. So, which of the statements is true about the circulatory system of the Daphnia magma? Okay, so it's single open. So the key point here is you should have applied the knowledge you have about insects, that they have these single open transport systems. So open means that the tissues are directly available to the transport medium that's going around the body. So the hemolymph goes directly to the tissues. It's not enclosed in vessels like it is in a closed circulatory system. And single meaning it doesn't go around the body twice. It doesn't go to their lungs, and then, because they don't have it in that same way, they have the spiracles in locusts. So the idea is it doesn't go round to the lungs and then back round to the heart and then back to the body again. That would be double, like in mammals. Okay, interesting sort of guessing game here. Where do you think the heart of the Daphnia is? Around here? Incorrect. Here. They're really interesting. The heart, you can't really see it very well. The heart of Daphnia kind of situated up here. You can often see lots of eggs in Daphnia quite clearly as well, which kind of mix it up, and often you see their legs under the microscope kicking around. But this is where the heart is, and it's quite a good experiment, and we quite often do it now, where you can put them under the microscope, you can give them caffeine or alcohol to speed up and slow down the heart rate. And it's really clear to see, it's a good one to do. Okay, next one. Which one is not involved in the primary? So really important in the sense of primary, that's the initial infection. Okay, so do T cells kill initial infection? Yes, they do. Do helper cells? Yes, they do. They secrete the interleukins. Or do we make plasma cells in the primary? Yes, we make them, but we don't make the T memory cells. Okay, so the answer would be A. Okay, so more on to the actual questions now. The Titicaca water frog. Again, this is another example where you're having to apply something you've learnt. So you've learnt about the Linnaean classification and binomial system. So you need to apply that to this question. So what is the genus? Okay, so remember the two words, genus and species. Now you have to write this out. You aren't ticking a box, but it's really important we write it out. Now you're not expected to write in italics. You don't have to do that. You do have to write the capital letter. So you should have written the word out with the capital letter. Really, really important that we don't also include the species. If you just copy, how does the examiner know which one it is? You have to make that really, really clear to them. So capital T, and the spelling that's correct in this case. It's not normally as important as other subject spelling. However, it's giving you the word. Oops, sorry, it's giving you the word here. Okay, so you have to get that right. Okay, next question. Really common question here. Students tend to mix this one up. Now, this is quite a nice version of this question. Outline the properties of water which makes it ideal habitat for an amphibian. Now, if I was an amphibian, I wouldn't be talking about an insulating layer underneath that much, although it does allow ice in the answer. But have a go at this one. Outline, so what we're doing for outline, we're giving the overall argument. Properties of water, which is a good idea for an amphibian. Okay, so kind of really important thing here, the first one which gets missed the most, which is a solvent. It means things are going to be dissolved in the water. Lots of smaller creatures need to get things directly out of the water. In this case, oxygen is quite often important for amphibians and fish as well. Really important one to get here, get right, not to mix it up with high heat capacity or specific latent heat. We're talking about high specific heat capacity. So what is it? So this is specific heat capacity here. So it's the energy required to raise the temperature of a substance per unit mass, which means how much energy is needed to make water, in this case, go hotter by 1 degree Celsius, and it's 1 kilogram. That's what specific heat capacity is. Now, you should know this from your GCSE. We do cover it again at A level. So what does that mean? That means that a lot of energy, because it has a high specific heat capacity, a lot of energy is required to heat that water up. What does that mean? That means the water is then going to be stable. So the change in water for an amphibian that isn't a mammal, it doesn't have the ability to control its body temperature the same way that we do. It doesn't have this vasoconstriction, vasodilation, all these mechanisms of sweating, etc., and hair. So it has to be able to use its environment to keep its temperature stable. So it needs to live somewhere where the temperature is stable. Or described means you could have just described the fact that the water temperature stays stable. It's not likely to change very quickly. You could also say that the water has quite a high density, which is useful, so the frogs will float on it as well, which is useful for amphibians. They don't want to be sinking. There's going to be a lot of energy expended if they're having to continuously swim every time they go in water. And then finally, you could say that ice is less dense than water, which I'm surprised they've allowed that. But still, okay, ice. The idea is, because you talked about density, ice is going to float on the top. Okay, so next one. Like all amphibian, frogs are able to absorb oxygen through the skin as well as their lungs. Suggests why is evolved unusually large folds of skin. Again, applying what you know. Have a go. So, break down the question a little bit. Suggest, I've got a couple of OCR definitions down here. So, I'll try and give you my version as well. So, offer an opinion for a particular course of action or an event or issue. You've also got give possible alternatives, produce an idea, put forward an idea or plan for consideration. Now that's quite confusing. These are both OCR definitions. This is an older version, this is a newer version. I'll link you some other command words as well. But in short, the way I see suggest is come up with a reason why something happens. Okay, and even sort of simpler than that is apply what you've already learned in a similar situation to this situation. So if you've learned about large surface area to volume ratio, in lungs, for instance, you taught about that at GCSE, but they have a large surface area to volume ratio, so that allows an increase of substances. Use that information to answer this question. Okay, have a go. Okay, so we've got large or increased surface area to volume ratio. Why is that useful? So this is the description. The explanation of that is that's going to increase the rate of oxygen absorbed. Okay, it tells us the context of the question is absorbing oxygen, so you need to make sure you reference back to the context of the question here. Is that the oxygen is absorbed? Okay, so here is an example here. You can pause it now if you want to read through some of the other command words. I've also linked on the next page. Here is another set of the newest version of command verbs that I could find for OCR, so you can follow that link. Okay, next question. Now I picked this one out. This was the question probably out of the whole paper in which students missed the most. I found when I was a student, if I saw something unfamiliar, I would find it more difficult, but I kind of demonstrate to you here that you don't need to panic if you see something you've never seen before. You've not been taught about what methyl ester is. Okay, so if you're getting and you're reading through this question, you're looking through and you're starting to think, what is this about? You don't need to panic, okay? So let's have a look at the question itself. So you've got describe and explain. Pretty straightforward for describe and explain. We know what we're doing for that now. Just sorting my pen out. Okay. So, we've got describe and explain. Okay, so we're first of all looking at the pattern of the melting points. Okay, so we need to be talking specifically about the melting points of the 3 methyl esters. Okay, so if we kind of look at the table here, it starts higher and it ends up lower, so that's pretty straightforward. So the first mark of this question is really easy to get. However, lots of students mix this up. Okay, you can't, there doesn't seem to be, if you're unfamiliar with methyl esters, an easy way to describe this trend here, but what you will notice is the formula. Okay, so you need to apply now the number of hydrogens. So as the number of hydrogens decreases, so you can't say in the table, you have to say as the number of hydrogens decreases, the melting point decreases. So that will get you the first mark. Okay, which lots of students mixed up. They just said as the table goes down, melting point decreases, which is not going to get you any marks. You have to comment on the differences in the formula itself, and you'll see they have the same number of carbon atoms. Okay, so next thing, explain. So why is that? Now this is where your sort of basic GCSE chemistry is going to come in here. Okay, so if I do a very crude version of some hydrocarbons, okay, bear with me, I'm using my mouse. So if I've got something here that has carbon and hydrogens and oxygens, you should be aware, it sounds a bit like a carbohydrate in biology. This isn't a carbohydrate, but the point is we've got some kind of structure here. Now, if we're keeping the number of carbons the same, but the number of hydrogens is decreasing, okay, that means there must be more double bonds, a bit like an alkane and alkenes in GCSE. There needs to be double bonds going in. So if I'm going from this first one to the second one, I'm going to have to put in some double bonds. Okay, so if I put in this double bond here, okay, however many hydrogens I have, I now have to, I get my eraser, I now have to lose 2 hydrogens because now each carbon still has 4 bonds, because carbon's in group 4 on the periodic table. Okay, so each carbon now has that. Now, there is our 2 hydrogens that have been lost. Okay, so what does that mean in terms of the melting point? Well, I've taken the actual images of the 3 methyl esters. So you've got them in order here. This is the stearate. And you can see when it's more saturated, without this extra double bonds, okay, it's a relatively straight chained molecule. This is a skeletal formula. These are the carbons and they just don't draw the hydrogens on. Okay. Now, if we go down the group to the next one, you'll see now we've got this double bond in. This actually changes the shape of the molecule, and if we put another double bond in, it's changing the shape of the molecule even more. So how does that affect melting point? Okay, well, what that means is if we had all of these nice straight skeletal formula chains there for us, there are more intermolecular forces between them, which means you're going to need more energy to overcome those bonds. So if we go now to the mark scheme, you can see, okay, let's go to this more complicated bit to start with. So the more kinks, the less saturated, the more double bonds there are in the chain. Okay, it means that they are less uniformly packed together, so less energy is needed to melt it. So if you've now got all these curved molecules that are not uniformly packed anymore, it means there is now less intermolecular forces holding them together, so that means they're going to be broken down more easily, so less energy, less heat is needed to make it melt. Okay, quite a difficult question. Take a second to read through that, but you should be able to get it. Okay, we're nearly done. I think this is getting to one of our last questions here. So, DNA nucleotides. So, nice and easy to start with. Do you understand what DNA is? Well, if you know what the word is, deoxyribonucleic acid, deoxyribose, that takes us to kind of the first answer. Okay, so what is this? Deoxyribose. Okay, and we can work that out that it's deoxyribose. First of all, there's 2 ways in the question. It tells us that it's a DNA molecule, okay, not an RNA molecule. Okay, so that tells us it's deoxyribo, and also we don't have the second OH group on here. Okay, name the bond, so phosphodiester bond. Okay, always use that phrase in OCR, phosphodiester, if you're unsure, and it's the bond between the deoxyribose and the phosphate group. Okay, so next one. Identify 2 similarities and 2 differences. Now it tells us really importantly in the question, so say 2 of each. Don't say 3 of one and 1 of the other, say 2. Between the DNA and the molecule of ATP. So similarities, well, first of all, they've got a similar kind of shape. You've got these phosphodiester bonds. Also, if you know what adenine is in ATP as well, so adenosine triphosphate, You know that adenine is also the nucleotide base, that's another similarity. And then the differences, there's some obvious ones. So if I show you now, this is the structure of ATP over here. The first kind of obvious differences are, first of all, you have these two additional phosphate groups here. And also, what I highlighted earlier, you now have in ATP, you have ribose. Okay, adenosine triphosphate, which has a ribose, not a deoxyribose as well. Okay, right, let's finish off with a couple of questions for a bit of fun. Mammals questions. To estimate the number of cells in an adult human, have a go. Write a number down or say it out loud what you think it is. So the most recent estimate, so this is I think 2017 this was, is 37.2 trillion cells inside of an adult human. So Homo sapien. Next one. Interesting question. What is the mass of the heaviest testicle in the world from the right whale? It's not the largest whale, it's just got the largest testicles. Have a go. And it is C, 500 kilograms, which means with two testicles it has literally a ton. Interesting creature. Question 3. I quite like these creatures, hooded seals. They lose massive amounts of mass when they have a pregnant little seal pup that comes out, and when they store on lots and lots and lots of fat, and they give up so much of that fat to get that seal which has just been born as fat as possible as early. So how much of their milk then is lipid? 60% of their milk is lipid. Huge amount. Right, okay, so that's it for today. I'll put up on the screen now, once I find the slide, the exam paper. So here's the exam paper and the link. So if you want any more help, email me. Just submit this. You don't need to email me your scores now. Just submit it on Show My Homework. I will set it as a flexible homework where you can just press submit. I know you haven't been able to beforehand. Okay? Thanks very much, guys. Bye-bye.